Welcome to Lab 9. So now that we have covered the bones and a lot of the biological aspects of evolution and all of that, we get to classification. And so classification, we might have talked about it a little bit when we were talking about race, but we really didn't get into how we classify beings, whether animal, human, ancestral, human, right, into their different categories. And so we're going to get into that a little bit, and this is really setting us up and transitioning us from the last chapters of biological aspects of anthropology to more the primatology and archaic humans aspect of this course. So this is a really good transition chapter. We're going to be learning how we divide the species up into their several different categories, and then we're going to apply that in the next chapter to how we have classified the primates. All right, so classification. There are different levels of classification that we have, and so this really started off in the 1700s with a Swedish naturalist named Carl von Linné, or Carolus Linnaeus. And he assigned all plants and animals to two levels of classification, the genus and the species. And so we might have heard of genus and species before. My first, I think, as far back as I remember, my first experience even hearing genus and species goes back to my first time watching Alice in Wonderland, where the flowers are asking Alice what she is. And, they ask her, she's like, well, I'm a girl. And she's like, I know what you're talking about. And so, then they say your genus, your species. And then they say in the genus and species of a weed. So genus and species is really how we categorize and we identify various species. And so there's levels of classification. Genus is the more general level of classification that includes multiple related species, and so we belong to the genus Homo, so Homo Sapiens. There's also Homo erectus, Homo neanderthalensis, and Homo habilis, and so on and so forth, right? Those are all under the same genus. Our species, though, is the most specific level of classification, and this is uniting all of the related organisms. So only members of Homo sapiens are part of Homo sapiens, right? Only Homo habilis are part of Homo habilis, and so on and so forth, right? So the genus name is Homo, species would be sapiens, right? So each type of organism has its own species, and species that were similar shared a common genus. Then you go up, more and more broad up that classification ladder, and you get the other categories. Carolus Linnaeus gave us our genus and species, and later additional levels of classification were added to incorporate broader levels of relationship. Today, the major levels of classification are based on Linnaeus' system, and they begin with the species as being the most specific classification. So similar species, like I said, are grouped into the same genus to reflect their relationship to one another. Then similar genera are then clustered into the same family. Similar families are clustered into the same order. Related orders are grouped together in the same phylum. And then this leads us then to the highest level of classification, which is the kingdom. And the kingdom is made up of the related phyla. Every known organism belongs to a group at each major level of classification. These groups then reflect how the organism is related to other organisms. There are also levels of classification that fall between these major levels in addition to that order, right? There can be a suborder or a subfamily or a superorder, right? So there's other levels of classification. These are the real main ones that you're going to have to memorize. So kingdom, phylum, order, family, genus, species. There is a mnemonic device that is useful for remembering them, or at least is how I memorized it when I first took biology. And so my biology instructor taught us King Philip came over for great spaghetti, right? So King Philip is kingdom, phylum. Came is class. I know class isn't on here. You're going to see it in the next one. So class over for great spaghetti is order, family, genus, species. right? So kingdom, phylum, order, kingdom, phylum, class, order, family, genus, species. Okay, so King Philip came over for great spaghetti. Another one that's more R-rated, I guess you can say, is King Philip came over for great sex. That's another one. So you can just change it around however you want. So whatever helps you memorize that, remember it in that order. King Philip came over for great spaghetti. So remember kingdom, phylum, class, order, family, genus, species. So to take a look at this, we are going down the taxonomic levels to us Homo sapiens, right? And the way that in my brain, the way that my brain works is I when I think of categories, it's always, does this have this or not, right? Does it have this one, trait, or doesn't it? And that's what you make, that's the easiest way to divide up a lot of the taxonomic breaks, even with our primates. And I will probably be talking about it in that way when we get to primates in the next chapter. So kingdom, we Homo sapiens belong to the kingdom Animalia, right? There are higher than kingdoms, right? I think the biggest breaks are between bacteria, archaea, and Animalia. So there's other ones, but kingdom, we belong to Animalia. And so what this means is that any species or any specimen, you could say, because we're not going to say species, any specimen that falls under Animalia is going to be a mobile, multicellular organism that consumes other organisms for food and develop during an embryo stage, right? So whatever falls under those class of characteristics is under the kingdom Animalia, right? We'll skip over, I guess I'll read all of them, but remember you want kingdom, phylum, class, order, family, genus, species, right? So kingdom Animalia, we belong to the subkingdom Eumetazoa, right? These are all major animals except sponges that contain true tissue layers organized as germ layers which developed into organs in humans, right? We belong to the phylum Chordata. This is the group of vertebrate. So chordata, think spinal cord, right? Group of vertebrate and invertebrate animals that have a notochord, right? That is the earliest form of a spinal cord that we see. And so any animals that have a notochord, this then becomes the vertebral column as you get more evolved outwards, you can say. So vertebral column in humans and other primates. Right, so kingdom, phylum, we have the subphylum Vertebrata. These are animals with vertebral columns or backbones. This includes fish, amphibians, reptiles, birds, and mammals. You narrow it down more to the superclass Tetrapoda. These are vertebrate animals with 4 feet or legs, including amphibians, birds, dinosaurs, and mammals. You go to the class Mammalia. These are warm-blooded vertebrate animals that produce milk for their young in mammary glands. They have hair or fur and specialized teeth. You narrow it down more to our subclass Theria, which is a group of mammals that produce live young without a shelled egg, including placental and marsupial mammals. You narrow it down more to order. We are from the order Primates. These are groups of mammals specialized for life in the trees with large brains, stereoscopic vision, opposable thumbs, grasping hands and feet. Further, we belong to the infraorder Anthropoidea. So this includes monkeys, apes, and humans. They have, in general, long life cycles and are relatively large-bodied. The superfamily Hominoidea. These are anthropoids including humans, great apes, lesser apes, and human-like ancestors. They have the largest bodies and brains of all primates. Then family is Hominidae. These are great apes, humans, and human-like ancestors. Our genus Homo, so group of hominids including modern humans, their direct ancestors, and extinct relatives such as Neanderthals. They are bipedal and have large brains. And then our species Sapiens. They are modern, ancestral, modern human, and they have culture, use language, inhabit every continent except Antarctica. So as you can see, going from the top to the bottom, you get more and more and more specific. So in the beginning, it's just mobile multicellular organisms, right? Now it's mobile multicellular organisms, right? But we also have tissue layers, right? Then it's mobile multicellular organisms with tissue layers that then have a notochord, right? Which becomes vertebral column. Then you narrow it down, it's multicellular organisms and, consuming other organisms, all that stuff, with true tissue layers or germ layers and a notochord and vertebral columns or backbones, right? So now you have to have all of these to belong to this. Then you go down, right, and I'm going to stop saying all these, but vertebrate animals then that have 4 feet or legs, including amphibians, right? Then you continue to narrow it down. Vertebrate animals with 4 feet, also warm-blooded, right? So you just keep adding more and more and more and more specific until you get to your genus and species, right? Specific, the most specific being species, right? So this is how we've classified humans. You just think of it as each level of classification gets more and more narrow. If you go upwards, right, from species to kingdom, you can then trace how we might be related, right? So one of the fun things that I like to think of, and I usually ask a class, is I ask them, if I were to ask you to guess, who is yeast more closely related to? And I give you the options between bacteria and humans, right? And you can take a minute, you can try and guess it yourself, right? I usually ask you guys, like, who is yeast more closely related to? Bacteria or humans? I'd say about 80% of the time, or every time I ask this, about 80% of the class will say bacteria. Because you think of yeast and they're these like unicellular organisms, right? And it seems like they have more in common with yeast. But bacteria belongs to that kingdom of bacteria, whereas yeast belongs to the kingdom of Animalia, I believe. And so because of that already classification difference between yeast and bacteria, right? Yeast is more closely related to us humans than it is to bacteria, right? So again, it's just something to keep in mind, right? You can see it as we go up and up and up and up, right? So why are we more related to primates than we are to, say, dolphins, right? They're both mammals, right? And we can see it because they belong to this group. They belong to this group. They belong to this group, and this group, and this group, and go higher and higher, right? Dolphins would come in, right, probably around here, right? Live young without a shelled egg, placental marsupial would be right about here, right? So it makes sense, right? Dolphins would branch out here, but if we're asking, a chimpanzee, right, they would branch out right around here, right? So actually right about here, right? So hominidae. So you just see, it just helps us see really how closely related or how distantly related or how we can group together organisms. So classification is really, really helpful for these purposes. So it gets to how do we name specimens? Right. So each type of organism has a 2-part scientific name, and this is called binomial nomenclature, and it's made up of the genus and the species. And so the way this binomial nomenclature, the rules for binomial nomenclature are very, very, very important. And I want you guys to make note of this and make sure you master how to write scientific names. And you have to do it in this section, because from here on out, if you write something, the scientific name of an organism, and you write it incorrectly, I will take off points, right? And that's not going to be a full point. It'll be like a quarter point or maybe a half a point, depends, right? But if it's like right away, it might just be a quarter point. But if it's like in another week or two, right, I will be taking off half points. So really make sure you know how to write the scientific name properly. And I'm going to go through it with you right now. So what are the rules? So again, each type of organism, two-part scientific name, binomial nomenclature made up of the genus and the species. These names tell us something about the organism. So Homo erectus means upright man. It tells us something about the organism. The names follow specific rules, particularly in terms of how they're written. So when we are writing the genus and the species, right, the scientific name, the genus is written first, followed by the species name. So it's always Homo Sapiens, Homo erectus, genus Homo, species erectus, right? The genus name is always capitalized, right? And the species name is always lowercase, always. There's no exceptions. Both words must be italicized if we're typing, right? So you guys are all typing, right? Because it's all on Canvas. So both words must be italicized. If you're writing by hand, and if for whatever reason you're writing scientific names by hand, you underline. Okay? Then we get to shortcuts, right? So you're not going to have to write Homo sapiens, Homo neanderthalensis, Homo habilis every single time. You can shorten them. So if you are referring to the same species repeatedly, right? Say you're writing a paper, right, for publishing, and it's a paper about Australopithecus afarensis. You can shorten it to capital A dot afarensis. And afarensis, remember, species is always lowercase, so afarensis will still be lowercase, even though there's a period there. If you're referring to the same species repeatedly, you can abbreviate the genus name to the first initial, followed by a period, and then you follow with the species name. That letter is still capitalized, and this can only be done if you have already written out the entire genus name earlier in the work. So in your homework, in your labs, when you are writing it out, the first time you type out, say, Panpaniscus, right? You have to fully type out Panpaniscus, right? And Panpaniscus is our bonobos. You fully type out Panpaniscus, and then if you refer to Panpaniscus anytime after that, you can then abbreviate it to capital P, period, and then Paniscus. And Paniscus is lowercase, right? So again, type it out the first time, and then every time after that, you can abbreviate. So if I were to do this here, right, and I had typed out somewhere Homo erectus, right, then I would type out H. erectus, right? Let me see if I can actually, let me see if I can, no, right. So if you need help with that, I can help you with it. We can do practicing. We can practice some together. You can just set up a meeting. Right. The other thing I have to tell you guys is if you're writing about two genera that start with the same first letter, you then abbreviate the genera to be the first letter capitalized and the second letter lowercase, followed by a period. So let me give you an example. If we have written an article about Australopithecus afarensis and Ardipithecus ramidus, we can abbreviate the two names, right? At some point earlier in the paper, we wrote Australopithecus afarensis, Ardipithecus ramidus, and then every time after that, we want to abbreviate. But if we were to just put A afarensis and A ramidus, we would assume, a reader could assume then that they are the same genus as well, but they're not. One is Ardipithecus, one is Australopithecus. So in order to avoid that, we would do AU, right? Capital A, lowercase u, period, afarensis. Then we would do AR, right? Capital A, lowercase r, period, ramidus, right? And then we would do it like that. So it's, again, after the first usage, you can then abbreviate to AU afarensis and AR ramidus. So that's something else to keep in mind. It's going to come up in the labs. It's going to come up in the quiz, most likely. Right? So that's how we write genus and species. Right? But how do we divide them? Right, now we understand the mechanism of how we write it, right? The rules for it. How do we actually divide species into the different categories, right? So it's difficult to define a species in the case of plants or microorganisms, right? It's a lot more difficult. Luckily, we're not going to be talking about that. In biology, we use the biological species concept. And this is often used to address the issue of delineating and differentiating our different species. So this definition is not universal, but it's the most practical definition used, particularly when you're looking at vertebrate animals. So according to the biological species concept, two organisms are from the same species if they can produce viable offspring, right? So that is the key thing here. They must be able to produce viable offspring. Two organisms then are from different species if they cannot produce viable offspring. So the question is, what are viable offspring? So in biology, viable offspring must be living and be capable of reproduction. So it's not enough that the two organisms can produce a living offspring, but the living offspring must also be able to reproduce in order to be considered viable. So living and capable of reproduction, that means it's a viable offspring. That means two organisms are from the same species. So here's an example, right? We're familiar with horses. We're familiar with donkeys, right? We have a horse here, donkeys here, right? And we're familiar with mules, right? Here, right? Which we know are the offspring of a horse and a donkey. And so what we have, and we know that horses and donkeys are separate species. They are not of the same species, yet they are able to have a living offspring, which is the mule. So how does this happen? Horses have 64 chromosomes. Donkeys have 62 chromosomes. So when a female horse and a male donkey mate, each will contribute half of their genetic information, right? That's basic biology. When mating occurs, half of the genetic information is passed on to the next generation, right? So the offspring of a horse and a donkey will receive 32 horse chromosomes and 31 donkey chromosomes. This combination can produce living offspring, right? We know that as the mule. However, the presence of the unmatched chromosome interferes with the meiosis cell division involved in producing gametes. And all male mules and most female mules are infertile, right? So they are no longer capable of reproduction. So despite the fact that the mule is alive, it is not considered to be a biologically viable offspring because it cannot reproduce. Therefore, donkeys and horses are separate species because their offspring are not viable. They are not fertile, right? So that's the biological species concept. Distinguishing species based on the biological species concept is challenging when you're dealing with extinct organisms, right? So it's very easy when we're talking about current organisms that we can see and we can they can try and mate, and if they produce a viable offspring, we'll say they're the same species. But it's difficult when we're talking about extinct organisms, as is the case with a lot of the organisms that we study in biological anthropology, right? Especially our paleo primates, right? So in these situations, we are trying to distinguish between paleo species. These are extinct species. And so we are dependent on the fossil record. We are then forced, right? So how do we do this? We're forced to use physical traits as indicators of the relationship between specimens. These physical traits will often reflect behaviors, things like diets and form of locomotion, as we'll see when we talk about diet and locomotion in primates, right? We can use physical traits then to determine if two fossils have both anatomical and behavioral similarities. So the more similar the fossils are, the more likely they are to be related. We can also take into account the geographic distribution of the fossils and their dates. So if a fossil is found in a specific region, let's say Argentina, right? I think that's where a lot of dinosaurs are found. And so we're looking in a specific area in Argentina. We are not going to try and see if a specimen found in, I don't know, China, we'll say China, is related in it by species to the specimen found in Argentina, right? Because they're not in the same geographic distribution. Another thing is we're going to look at the dates, right? If we're looking at a specimen that is at a higher strata, right, one that's more recent, compare it to one that was found, 40. I'm not really good at this at the metrics of it. Let's say not 40. Let's say like 3 or 4 layers down, right? I'm not going to give an actual number. 3 or 4 strata downwards, then, which is an older specimen, we're not going to try and see if we might, but we won't really go. Our first assumption won't be that they are related, the similar species, right? We won't assume they're similar species, because between each strata there could be many thousands of years separating them out. Right, so again, we take into account geographic distribution of the fossils and their dates, as well as then the physical traits as indicators. So how do we do this, right? There are 2 types of trait similarities that are found between biological organisms: homologies and analogies. In both cases, we are dealing with similarities that are shared between organisms. However, shared traits may come about for different reasons, and homologies and analogies are the results of 2 different processes. So we're going to first start with analogies, and then I'll go into homologies. So analogies are traits that are similar across organisms due to similar adaptations. So these traits, these analogies, are the result of homoplasy. Homoplasy is the evolutionary development of similar traits in unrelated organisms. So this can often be the result from circumstances as similar as the diet and environmental conditions. So let me give you an example here. If we look at a colobus monkey, which is what we have here, and a cow, right? A colobus monkey and a cow, right? We see that they both have complex stomachs, right? This is an analogy that they have. They have complex stomachs. Both of them eat a lot of plants, which are difficult to digest and require specialized digestive tracts. But when we look back at the common ancestor of colobus monkeys and cows, we see that they did not have a complex stomach. So the trait, the fact that both of them have complex stomachs, that trait evolved separately in the colobus monkey and the cow lineages. Thus, the fact that both the colobus monkey and the cow have long digestive tracts is the result of similar dietary adaptations rather than something that was inherited from a common ancestor. Right? So this is an analogy. When we classify organisms, right, we try to group them based on their biological relationships to one another. So this is why biological anthropologists have to distinguish between analogies, which can be misleading, right? If we were just to take the colobus monkey and the cow and say they both have complex stomachs, therefore they must be closely related, right? That is very misleading, right? Because if we look back to the closest shared ancestor of a colobus monkey and a cow, we see that they don't have that complex stomach. So it's not something that they inherited in their lineage from a shared common ancestor. So analogies can be very misleading, right? So we distinguish between those analogies and homologies. And homologies here are actually helpful for classification. So homologies are traits that are similar among organisms that share common ancestry because they were passed down from a common ancestor to its descendants. So this is something that we share in common from that last shared common ancestor and has been passed down. These are traits that reflect real ancestral relationships between organisms. So these traits are much more helpful for classification than the misleading analogies. So there are different types of homologies, and some of these homologies might be more helpful than others, right? So for the most part, we're going to be talking about homologies more than analogies, right? Just know that analogies come about from, similar adaptations to similar circumstances, whereas homologies are actually ancestral relationships. So the types of homologies. So we have ancestral traits and derived traits. And then within derived traits, we have two types. We have shared derived and unique derived. So ancestral traits, or plesiomorphies, are traits that are inherited by two organisms from a relatively distant common ancestor. So let me give you an example. So humans and ring-tailed lemurs both have mammary glands. This is a shared trait that was found in the common ancestor of humans and lemurs. This trait is useful in that it supports the idea that humans and ring-tailed lemurs are somehow related. However, this is a trait found in all mammals and can then be traced back to the last common ancestor of this entire group of animals. So it's not particularly useful then in helping us actually distinguish between humans and ring-tailed lemurs. For this reason, biological anthropologists often then will focus on the other kinds of homologies called the derived traits, right? Derived traits or apomorphies are traits that are modifications of ancestral forms. So there are two types of derived traits, like I said, the shared derived traits and unique derived traits. So shared derived traits, synapomorphies, are traits that are modified traits shared by two different groups of organisms. So I give you an example. I think the examples help us understand this a little better. So apes and old world monkeys have the same dental formula. This is a modification of an ancestral trait, a dental formula that was slightly different. Thus, it is a derived trait because so the fact that it's a modification of an ancestral trait makes it a derived trait. Because it is a modification that was shared between the two organisms, it is a shared derived trait, right? So shared derived traits are traits that are modified traits shared by two different groups of organisms. So then we have unique derived traits. These are modified traits unique to one group. So in this example, we have humans. Humans have an unusual pelvis that is very round and bowl-shaped. This is a modification of an ancestral pelvis that was flatter. Thus, it's a derived trait. Because the modification is unique to humans, it is a unique derived trait. Researchers today will also use genetic analysis to help us classify organisms. An anthropologist may examine DNA or amino acid sequences to look for similarities and differences between organisms as well. As with physical features, genetic similarities are used to infer ancestral relationships. And the more similar two organisms' genes are, the more closely related they're assumed to be. Right? So, homology versus analogy. Right? So this is an ancestral trait, the fact that they both got mammary glands. Right? Here is our shared derived trait. Right? So we have an old world monkey under an ape, sorry. This is an ape, not an old world monkey. We have an ape here, and then we have B, an old world monkey. They both have the same dental formula. This dental formula is a modification of an ancestral form with more teeth. So it is a shared derived trait. I'm trying to think. 3, 2, 1, 2, 3. So the dental formula here, so I was just looking at the dental formula. The dental formula here is 2, 1, 2, 3. And same thing here, 2, 1, 2, 3. It's the same dental formula, right? This dental formula is a modification of an ancestral form with more teeth. So shared derived trait. Then we have our unique derived trait here. And so we have a human pelvis here and a primate pelvis here, right? This is a modification of an ancestral form that is unlike the pelvises of our other living primates, right? This rounded pelvis is a unique derived trait, right? So keep those in mind. You're going to have a lab exercise on that. So processes of classification. So the classification of life is called taxonomy, and this forms the basis for systematics, which is the study of the relationships between organisms. In modern systematics, biological anthropologists use two primary approaches to classification, and we use evolutionary systematics and cladistics. So evolutionary systematics is the first and longest running approach to classification, and this approach emphasizes both our derived traits and ancestral traits as useful tools in classification. Whereas cladistics is the more recent approach to classification, and this approach emphasizes derived traits and de-emphasizes the ancestral traits. So proponents of this view argue that the more derived traits are more useful in distinguishing groups. So both approaches to classification emphasize homologies, and they really avoid those misleading analogies. The difference, though, between the two is the type of homology that is emphasized. So if a researcher has just used cladistics only to identify the biological relationships among a set of primates, the easiest way to share that classification is to create a diagram of the relationship. So really we're focusing here on cladistics. So the type of diagram used to represent cladistic classification is what we call a cladogram. And so a cladogram, and you might have seen a cladogram before or a version of it. A cladogram is the diagram used to represent cladistic classification. It lists every organism included in the classification at one level, right? It's usually at a row along the top, right? They're all at the top. The organisms are then connected by a series of lines, right? As you can see here. And then the fewer the lines between two organisms, the more closely related they are. So here we have, and I think I have it bigger. No, I don't. Here we have 4 primates, right? A black and white colobus, a red colobus, a chimpanzee, and a human. And these 4 primates are branched into 2, right? 2 types of colobus monkeys connect to each other at one point, at point A on the diagram. This point represents their common ancestor, and it shows that they are more closely related to each other than they are to the other primates in the cladogram. Similarly, our chimpanzee and human are closely related to each other, and so they also connect to each other before they connect to any other primates, showing their common ancestry, which is point B. Right? So here we have point A, which is the last common ancestor between black and white colobuses and red colobuses. Here we have point B, which is the last common ancestor between chimpanzees and humans. All 4 primates, black and white colobus, red colobus, chimpanzees, and humans, are all distantly related to one another. And that is why they all connect at point C to show their common ancestry. Right? This is the last common ancestor of all 4 of these species. Right? So that is a cladogram. So cladograms are constructed through a process of cladistic analysis that begin with identifying potential homologies. The presence or absence of these traits or characters are then identified and recorded in a matrix that includes each organism under investigation. So, if we go back here to the cladogram. So when you look at our sample cladogram here at the top, we can see how the traits studied helped the researcher to both cluster and distinguish the different organisms. All the organisms have a narrow nose, right? So this is our sample data matrix. We're going to go based off of these 4. Narrow nose, multicolored fur, broad chest, or fur-covered body, right? And that's how we separate them out. So all of them have a narrow nose. Black and white colobus, red colobus, chimpanzee, and human. This is an ancestral trait then that demonstrates that they're all related to one another via a common ancestor. And then we look at the multicolored fur, right? And here we see that the black and white colobus and the red colobus have multicolored fur, and the chimpanzee and the human don't have multicolored fur, right? So this is a shared derived trait then that indicates that these 2 organisms are more closely related to each other than they are to the other organisms. Similarly, if we look here, right, we look at broad chest, we see that the black and white colobus and the red colobus do not have a broad chest, whereas the chimpanzee and the human do have a broad chest, right? This, again, is a shared derived trait that reflects their close relationship to each other. Finally, the human seems to be unusual because it is the only organism here, right, that does not have fur covering their entire body. So this is a unique derived trait that distinguishes the human from even its closest relative, the chimp, right? These 4 traits can be placed on the cladogram to show which organisms have each traits, right? So here we have here. So human, right, they have no fur. Chimpanzees, red colobus, and black and white colobus all do, right? So here, this point B, this break. If you go farther back, we see that chimpanzees and humans both have a broad chest, but we know that our colobus monkeys do not, so it's up here, right? So broad chest, we have these 2, right? Multicolored fur, though, here is where we have our colobus monkeys, right? And then our narrow nose right here, all of them have it. So each trait is placed below the point that represents the common ancestor of all organisms with that trait. This signifies that all the organisms above that point, right, above this point or above that point or above this point, all the organisms above that point have that trait. Another type of diagram can be used to show evolutionary relationships, and that is the phylogram. It's a little bit more complex than the cladogram. Really focus on the cladogram for the most part. So a phylogram, really quick, is similar in style and meaning, but it accounts for the amount of change. So usually we're talking about time. And what it does, it'll vary the branch length. So there might be more length between a chimpanzee and a human versus the length between the colobus monkeys, right? Both types of diagrams are used by biological anthropologists to convey important information about the relationship among organisms, right? So the longer the branch in a phylogram is used to indicate a greater amount of change, therefore a longer time span since the split between the 2 organisms. And both of them, the cladogram and the phylogram, tell us which species are closely related to one another and how other relationships extend beyond the different pairs of species. So I want to just go back really quick to show you this, to show you and show the reason why the data that you use, right, the classifications that you use really matters. Because if we were only, let me see, if we only looked at, if we took out broad chest and multicolored fur, and we were only looking to see narrow nose versus fur-covered body, right, we would then branch the chimpanzees with the black and white colobus monkeys, and then humans would be on their side. So it really, really is important that we look at traits that can really help us determine where to put specific species, right? Yeah. Go back. So process. So with this science, as in most science fields, there is often some kind of conflict and disagreement. So there's often disagreement about how to classify organisms, particularly when dealing with fossils that can't be genetically tested or directly observed for interbreeding potential. So we have 2 groups of researchers, right? Some are considered lumpers because they tend to group or lump organisms into a few large groups. Others are splitters, right? Our lumpers tend to emphasize the similarities between our organisms and will often interpret differences as minor variations within the group. Our other researchers are called splitters because they tend to divide or split organisms into numerous smaller groups, right? They really like to classify them as much as possible. Splitters tend to emphasize the differences between organisms and will often interpret the differences as substantial variations between behavior, ecology, and adaptation of separate species. And this lumpers versus splitters is going to continue to emerge, especially when we're talking about the human lineage. So lumpers and splitters often result in two very different interpretations of the exact same evidence, which is why there's not always agreement on how to classify organisms, specifically or more particularly our fossil species. At the same time, there are additional points of disagreement and departure in classification schemes. So, an example, if an anthropologist chooses different traits to analyze, right? And this is going back to the earlier example again. We'll go back to this. We choose 4 traits to analyze, but any other traits could have been used, right? The traits we use will influence the conclusions we come to. If we had not chosen that broad chest trait, we might not have recognized the relationship between the chimp and the human. Without that connection, we might have classified the chimp as more closely related to the black and white colobus and the red colobus because they all have fur. In some cases, it's difficult to distinguish traits, as is often seen in genetic analysis. So imagine then, let me go back here. Imagine then, for this example, if a researcher has sequenced the DNA from 2 organisms and is not looking for stretches of DNA that are similar and thereby indicate an ancestral relationship between organisms. So this task requires the researcher to align the stretches of DNA from both organisms and then look for overlap. So because overlap in bases may occur at multiple points, the anthropologist must make a judgment call and then decide which alignment is accurate and shows a meaningful similarity between the DNA. So that's what we have. Oh, I don't have it here. In your slides, if you look at the next slide in your slides, you'll see the 2 lines of DNA. Other anthropologists may make different judgments about where the DNA strands are similar, so they might come up with different conclusions about the relationships between organisms. So it's important to remember that the process of classification is full of choices, and those choices may lead researchers to different conclusions. Overall, the perspective of the researcher, the traits they use, and how they interpret those traits will then affect the final classifications made by the researchers. Right. So that's the end of lab 9. This really sets us up again for the next lab, which is on the taxonomy and the classification of our living primates. That is a more in-depth chapter, right? So really keep the homologies and versus analogies in mind. Keep in mind, and that's really the analogies, or not the analogies, the homologies are really the ones that you're going to want to keep in mind in order to help us identify and separate out the different species that we are going to be talking about in the next lab.